Is that like an experiment or something? All right. For those that are Echo 360 users, Austin couldn't be here today, and so nobody's watching the store. And if you've got any questions, just put your hand up today. So last week we introduced the topic of protein transporters and went over the group classified as transporters. Today we're going to look at the ion channels, which is the other big group. And the cartoon on the left is sort of a generic representation of an ion channel, one that you would find in bacteria. And it's meant to indicate that this is a multi-subunit protein. So you've got several individual polypeptides that are embedded in the membrane, and they are collectively forming this channel structure. That is a protein in which you've got an aqueous pore here, and then you've got another portion of the protein that actually interacts with the membrane itself and is going to have nonpolar groups here oriented toward the interior of the membrane. Ion channels, remember, are different than the transporters in that they don't have specific saturable binding sites for the solutes that they allow to pass. And then second of all, instead of having two gates, an inner and an outer, which are created really by conformational change in the protein occurring, the ion channel is going to have a single gate. And what it does is it restricts passage to ions that have a certain radius to them and then have a certain charge. So this particular guy is a potassium channel, and in bacterial cells, potassium is going to be present in higher concentration in the cytosol than it is out in the environment in most cases. So which way is the potassium going to want to move if the channel is open? Going to want to go out. That's correct. Down its concentration gradient. Does that make this active or passive transport? Passive, that's right. And of course, in the water environment of the cytosol, these potassium ions, which have a positive charge, are going to be surrounded by a shell of water molecules, because water is a dipole. It has both partial positive and partial negative charges, and so those partial negative charges are going to orient themselves toward the ion. When the ion enters the vestibule, or the opening of the channel, it's going to move into a portion of the protein where that channel gets narrower and narrower. And eventually it gets so narrow that with its shell of water molecules, it couldn't actually get through. But the R groups here that are poking out from the protein subunits are going to actually pull those water molecules off. That is, the water molecules are now going to interact with the side chains on the amino acids instead. So it's pretty much the dehydrated or naked ion that goes through the gate of the channel. Then here on the other side, there are more water molecules. And of course, as soon as they see that positive charge poking its head out through the channel, it's like, I want some of that, and they're grabbing onto that, which is going to tend to make this ion want to keep moving in this particular direction. So this channel, this gate, acts as a selectivity filter. By having a particular width to it and a particular charge configuration around it, it's going to only let certain things go through there. It's only going to let, in this case, positive ions go through because, I mean, what's going to attract a positive ion? What do you think? Negative charges. So there's going to be amino acid side chains here that are negatively charged. What if a chloride ion got in there? Cl minus. What do you think is going to happen? That's right. They would be the same charge, they'd be repelled, and so it would bounce right back out here. And so there is a certain degree of specificity to an ion channel, but it's not absolute. In fact, we take advantage of that in the lab. You can take ions that are in the same period of the periodic table and get them to go through a channel that functions in the cell as, say, a potassium channel, but will let certain other ions through, things that aren't biological ions, but they're the right charge, right general shape. And they don't go through as well, but they'll still go through. And again, we can make use of that. So again, this is just sort of a schematic of what a channel looks like and how it works. This is a structure or a cross-section of a structure of a real ion channel. This one isn't a potassium channel, it's a calcium channel. And it goes by the name of ryanodine receptor 2. Ryanodine is a toxin that happens to bind to this channel, so that's how the channel got its name. But its job is to let calcium, which in muscle cells is kept in the endoplasmic reticulum, or in muscle we often call it the sarcoplasmic reticulum, until the muscle gets a signal to contract. And when it does, this channel will be triggered to open. And once again, there's a narrow selectivity filter here that will allow calcium ions to pass through this pore, strip the waters off them here, and then allow them to flow out with their concentration gradient. Because the calcium, in the absence of a contractile signal, is kept high here, it's low here out in the cytoplasm. And so this guy's job is to let out the calcium, which is going to trigger muscle contraction when it's time for that to happen. And then there's a pump in the membrane, a calcium pump, that will, after contraction cycle has taken place, pump all that calcium back here into this space. And it will stay there until this calcium channel opens up again. That brings us to a subject that really throws people, and that's membrane potential. We're talking about electrical potential here. Does anybody know how you define electrical potential? Somebody with a little bit of an electronics background. Yeah. Okay, it's a voltage difference between 2 points. When we say the term potential, what are we specifically talking about? Does anyone know? Pardon? Okay, it's not the way electrons would move. That's what you would think. That's certainly what I always used to think until I did some reading. It's something else. The direction that something would move, what? What kind of charge? Positive charge. It's the direction a positive test charge would tend to move. And the more the positive test charge would want to go in a particular direction, the higher the potential. Biological membranes almost always have a potential across them. That is, a positive test charge would want to go in a particular direction. And the direction it typically wants to go is from outside the cell to inside the cell, if we're talking about the plasma membrane. Now, how do you get this situation? Well, here's a hypothetical cell membrane. This is completely hypothetical. And there are all kinds of different charge species that you'll find inside and outside of cells. If we just focus on the inorganic ions, there are things like sodium, potassium, protons, calcium, magnesium. These are all cations. But there are also anions. There's chloride, bicarbonate, the hypo and hyperphosphate phosphate ions. All of these things carry negative charge, and there are some of both on either side of the membrane. In a situation where there were an equal number of positive charges and negative charges on either side of the membrane, will we have a membrane potential? What do you think? Yes or no? No, because a positive test charge wouldn't tend to want to go in either direction, because the total charge on either side of the membrane is balanced. What if now there was some way for some charge to leak across that membrane, to go from one side to the other? And so, again, hypothetical situation. We've still got our membrane barrier here. Remember, lipid bilayer membranes, charged species cannot just diffuse across them. They're going to need some kind of a protein to help them out here. But in our hypothetical situation here, some positive charges have somehow gotten across the membrane from this side to this side. That means there is a slight excess of positive charge on this side of the membrane relative to this one. Now, which way would a positive test charge want to move if it had a direction for doing so, if it had a pathway, I should say. Would it want to go this way or this way? Yeah, it would. It would, whoops, I hit my button. It would want to go to the right. And this is, in fact, what happens in cells. And it happens because there is a type of channel that you find in plasma membranes called a potassium leak channel. Potassium leak channel is a species that fluctuates constantly between its open and its closed conformation. So when this leak channel is closed, the gate is shut, no ions can move through the channel, and the balance of charges stays the same on both sides of the membrane. But when this thing is in its open conformation, potassium ions can move in the direction that their concentration gradient pulls them. And as it happens, the concentration of potassium is kept higher in the cytosol of cells than it typically is outside of cells. So which way are the potassium ions going to want to move? Down or up? Based on their concentration gradient, they would just want to move up. That's right. But it's not all about just their concentration gradient, because they're charged. And therefore, how much potential there is for them to move across that membrane is affected by how much positive charge there is on either side of the membrane. He said that, yeah, there's more potassium on the inside of the cell, so definitely it wants to go this way. And if all we were focused on is the potassium, we would say, yeah, you know, and those positive charges would want to get away from the other positive charges, and so they'd want to go, wow, right through that channel. But the thing is, there's lots of other ions around. And in fact, there is another reason that they wouldn't want to go this way from a charge point of view, and it has to do with the reason that there's more potassium inside the cell and out, and we talked about it last week. Anybody remember what it was? It was one of the transporters we talked about. Somebody said it real loud. Can you say it again louder so everybody hears? The sodium-potassium pump. Everybody remember that guy? This was the primary active transporter, and to be a primary active transporter, you've got to capture an energy source, in this case ATP, and you've got to use it to do work. You've got to move something. And the sodium-potassium pump hydrolyzes ATP and uses that energy to move ions against their concentration gradients. And what it does is it moves 2 potassium ions from outside to in. There's the explanation for why there's more potassium inside the cell than out. But it moves 3 sodium ions out. 2 positive charges in, 3 positive charges out. So what does that mean? It means there's more positive charge outside the cell than there is inside the cell. So this is going to counteract the tendency of the potassium ions to want to get through that leak channel and get away from the other positive charges of potassium ions, because when they get out here, it's like, well, there's a whole bunch of sodium ions out there, and they're positively charged, and that repels them. So you've got 2 forces here. You've got an electrical gradient that wants to move a positive charge this way, and then you've got a concentration gradient for potassium that says, you know, diffuse from high concentration to low concentration if you got a pathway through the channel here, and that wants the potassium to go this way. The 2 of them have to balance each other, and they do. And that determines what the equilibrium potential of the ion is. That is, how much potassium is going to move in what direction. And so what actually happens is that potassium does leak from inside to outside, but not as much as you would predict if you were just putting on blinkers and only looking at the potassium. If that were the only ion around, then we would say, yeah, man, the potassium would just rush out until the concentration was the same on both sides of the membrane. But that's not the world that it lives in. There is, yes, a lot more potassium in here, a lot less out here, but there's more positive charge total out here than there is in here because of the activity of the sodium pump. So some potassium leaks out, but not as much as you would predict. And we can predict exactly how much is going to go through the membrane under any conditions by using something called the Nernst equation. And before you ask, no, you don't have to memorize it. And I'm not even going to ask you to do a calculation on the exam with it. But the Nernst equation lets us predict what the equilibrium potential for any given ion is if we know what its concentration outside the cell is and what its concentration inside the cell is. Now, the rest of the equation here, this fudge factor, if you will, you'll see represented different ways. This is kind of a simplification of a more complicated expression that involves natural logarithms, but it works out to 62 times the base 10 log of this ratio here. So if you can measure the concentration of potassium on one side of a membrane and the other, you can figure out what its equilibrium potential is. That is, if the potential were this, you would have no net movement of potassium through the channel. But these leak channels explain in part why there is an excess of positive charge outside the cell relative to the inside. The sodium pump explains the rest of it. 3 positive charges in sodium out, 2 positive charges in sodium in. So what that means is that relative to the outside of the cell, the inside is slightly negatively charged. Now, the problem with that is quite often you'll see diagrams of membranes. And you say, okay, this line, this represents the membrane. And on one side, they'll write plus, 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 plus. And on the other side, they'll write minus, 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 minus. And what is the thing that you naturally assume from that is that there's nothing but positive charge outside the cell and nothing but negative charge inside the cell. That's not correct. It's just there's an excess of the one over the other. And so by definition, the inside of the cell is negatively charged relative to the outside. But there's positive and negative charges on both sides of the membrane. How do we know what the concentrations of ions are on both sides of the cell? How do we actually measure that? How do we know when current is flowing through an ion channel? Well, there's a very neat little trick called patch clamping, which is illustrated here. And if you talk to an electrophysiologist or go into an electrophysiology lab, they can tell you all about this because it's the bread and butter of what they do. But in patch clamping, you take a very, very, very thin capillary. It's a glass capillary. And you can see the width here is 1 micrometer. That's about the width of a bacterial cell. And under a microscope, you actually push this against the membrane of a cell. And you apply a little bit of suction, very, very gentle suction. I actually don't know how it's done these days. In the old days, people would actually have a tube hooked up to their mouth, and they would just suck very gently like they were sucking on a straw. You don't want to suck so hard that you bust the membrane of the cell, but you just want a little bit of suction so that you pull the membrane tight against the opening of this glass capillary. And the opening is so narrow that only 1 or 2 ion channels are going to be present in the portion of the membrane that you have got stuck in the end of this pipette. And then you've got an electrode here in this pipette. And you got another one that is on the inside of the cell here. And that creates a circuit here, and you can actually measure the flow of electric charge anytime that channel opens. And you can record that on an instrument, and the output looks like this. Every time the channel opens, boom, there's a spike of current. And then channel closes, boom, it drops back down to baseline. And then channel opens again, boom. This is called single channel recording. And so we can actually measure the opening and closing of individual channels. And then using a tool called an ion-specific electrode, we can measure the concentrations of an ion such as potassium in the cytosol and outside the cell. So we can, in fact, plug in values into that Nernst equation that let us figure out what the equilibrium concentrations for particular ions are. The gate in a channel can be one of several types. And that ryanodine receptor I showed you before, the calcium channel that's found in heart muscle, that is a ligand-gated channel. We talked about proteins and ligands before. What's a ligand? It does, okay, that is an example of a ligand. Substrate binding to active site is an example of a ligand. But the definition of a ligand is more general than that, because substrate is something specific. It's referring only to one kind of protein, an enzyme. And a substrate is a ligand that the enzyme can actually mediate a chemical reaction with. But the term ligand is more general than that. What's it mean? Anybody remember? Yes. Something that can bind reversibly and specifically to a particular site on a protein. That is absolutely right. And so the ryanodine receptor is a ligand-gated channel for calcium. And that means there's some molecule, there's some substance that binds to the ryanodine receptor and causes its gate to open. And you know what that substance is? It's actually calcium, the same ion that goes through the channel. So some calcium has to come in to the cytosol through another route, bind to the ligand site on the ryanodine receptor. That triggers it to open, and then that big mess of calcium that is sequestered in the sarcoplasmic reticulum or endoplasmic reticulum, whichever term you want to use, rushes through the channel. Other types of gates are voltage gates. We'll look at those when we look at nerve function. And in a voltage-gated channel, the membrane potential around the channel has to change. That is, the difference between the amount of positive charge on one side of the membrane and the other has to change in some way. And when it does, that will cause this channel to open up. Specifically, what has to happen is a phenomenon called depolarization. Membranes are normally polarized. That is, there is a gradient of electrical charge across the membrane because of the activities of the sodium pump and the potassium leak channels. And that makes the charge a little bit more positive on one side of the membrane, the outside of the membrane, than it is on the other side, the cytosolic or inside of the plasma membrane. If something causes that gradient to be collapsed or even reversed, that is, we go from a situation now where it's positive outside relative to inside to one where it's negative outside relative to positive inside, that will trigger the opening of a voltage-gated channel. And that's how nerves work. Ligand-gated channels can come in two flavors, one in which the ligand binding site is on the outside of the cell, and one where it's on the inside of the cell, shown over here. And then finally, maybe most interesting, are the mechanically gated channels. These are exactly what they sound like. There is something that the channel protein is attached to that can tug on it under certain conditions. And when force is applied, when that tug is felt, that mechanically gated channel opens up. And only while that force is being applied do you actually allow ions to flow through this channel. Here's a real good example of a mechanically gated channel. It's in your ear. And so most people have seen a diagram of the human ear at some point. You've got the outside portion of the ear, you've got the auditory canal, then you've got the eardrum, and then behind that you have a space, middle ear, that's filled with fluid, and that fluid is connected to a structure called the cochlea, which looks kind of like a snail here. And within the cochlea, there is going to be something called the organ of Corti. And in the organ of Corti, there are cells that have on them stereocilia. Now, in the lab, we're looking at organisms, tetrahymena, that have motile cilia that help the organism swim. Stereocilia are also composed of the same basic structures as motile cilia, but they don't move. Rather, they are sort of antennae or sensory structures. And so you've got these extensions of the plasma membrane that are held in place by microtubules, and they are sticking up, and they can actually interface, if you will, with something called the tectorial membrane here in the organ of Corti. When sound waves enter your auditory canal and they make the eardrum vibrate, that sets up vibrations of the liquid inside the middle ear, and that causes vibrations to make the tectorial membrane move. And when it moves, it tugs on these stereocilia. And the stereocilia have in their plasma membranes mechanically linked ion channels. And they are tethered by a linking filament to points on other stereocilia. And so when the tectorial membrane moves back and forth, it makes these things bend. When that happens, mechanical tension is applied to this channel. And that causes it to open, and when it opens, positively charged ions, sodium ions in this case, rush in through the channel. And that is going to initiate nerve signaling, and that is going to go to your brain, and that's going to tell you, hey, I just heard something. So it's all based on mechanical channel gating. Let's look at a voltage-gated channel now. So this is the plasma membrane in gray here. Here's a channel. It's a channel for sodium. And normally, concentration of sodium is higher outside the cell than inside the cell because of the activity of the sodium pump. And there's also a slight excess of positive charge outside the cell relative to inside. So if you opened up this sodium channel, where's the sodium going to go? It's going to come rushing in down both its concentration gradient and its electrical gradient. So in this case, we've got the electrical gradient and the concentration gradient working in the same direction. So if you were to pop this guy open, sodium would come pouring in to the cell. What's that going to do to the membrane potential? It was, with the channel closed, positive outside, negative inside. What's going to happen if you open the channel and a whole bunch of sodium rushes in? It's actually going to flip-flop. It's going to go from positive outside, negative inside to positive inside, negative outside. That's exactly what is going to happen. So here's our inactivated channel. Now something is going to cause a disruption of the normal membrane potential. Going to change the amount of positive charge on the outside relative to the inside. That's going to trigger this channel to open. And when it does open, sodium is going to whoosh right through that channel, and our membrane is going to become completely depolarized. That is, you're going to completely reverse the distribution of charge on the two sides, because now you got all this sodium inside. This voltage-gated sodium channel has a portion of its structure that is unstructured. It's not in any type of defined secondary structure. And it has a domain here that can swing on this mobile tether, if you will, and close up the opening to this sodium channel. Now, note that this is the same as closing the gate. Here's the gate here, or actually down here, I'm sorry. This is not closing the gate in the same manner that it was closed here. But it does block the entrance to the vestibule of this channel. So sodium can't get through anymore. And so as soon as the membrane becomes depolarized, this thing swings in and blocks it. So all of a sudden you stop the sodium from rushing in. Now, what's going to happen? Well, remember, there's all these other transporters in the membrane, and they're still doing what they're doing. And we talked about the sodium pump. So a bunch of sodium rushed into the cell. What's the sodium pump going to do with it? It's going to pump it back out again. And as it pumps it back out again, our membrane potential is going to begin to recover. That is, it switched from being positive outside, negative inside, to now being positive inside, negative outside. But the sodium pump starts moving all the sodium back out again. We're eventually going to get back to this situation here. And when that happens, our channel is going to close. That is, it's going to switch from its inactivated configuration here, where the tethered domain was blocking the vestibule to the channel, now to its closed configuration, where the gate is actually shut and nothing can get through it. And now we can go through another cycle again. That is, something can depolarize the membrane, and we haven't talked about what the something is yet. Something can depolarize the membrane, channel can open again, sodium can rush in, then that causes the tether to block the vestibule, and no more sodium can get in. The channel's inactivated. The period during which You're in this state here. It's called the refractory period. The channel cannot be reopened at that point. But then you will eventually pump all that excess sodium out of the cell through the action of the sodium pump, and this guy will return to his closed conformation, and we're back where we were. That's how nerve signaling works. So what you just saw is how you form an action potential. An action potential is an all or none depolarization of a membrane. That is, it's not a little bit depolarized. We didn't change from being, well, factor of 10 positive outside to inside to maybe being factor of 8 or something like that. We completely flip-flopped the polarity on our membrane by throwing these sodium channels wide open and letting all that sodium run down its electrochemical gradient into the cell. When an action potential is formed here, you have actually, in essence, created a type of electrical current. Now, in wires, in metal wires, we think of electrical current in terms of moving electrons, and that's what they are. In biological circuits, which is what nerve cells are, we're talking about the movement of ions, not electrons, and quite often talking about the movement of positive ions. And so here's our membrane at its resting state. Some stimulus comes along, and that causes a partial depolarization of the membrane. Not complete, but partial. That is, that charge ratio changes a little bit. And if that change becomes great enough to reach what's called the threshold potential, then those voltage-gated sodium channels open up. And when they do, boom, all of the sodium comes rushing in, and the potential across the membrane goes from being negative, minus 40 millivolts, to being positive, almost positive 40 millivolts. Just complete reversal of the situation that we had before. Then our open channels become inactivated. That happens as soon as depolarization occurs. That causes this tethered domain to swing into place, so no more sodium can get out anymore, so you can't get any more depolarized. And then the sodium pump takes over and begins moving the sodium out, and the membrane potential eventually returns to normal. Now, that's what happens if you're looking at a single sodium channel. Imagine you have a whole bunch of voltage-gated sodium channels, and you've got them spread all along the length of a nerve axon. We have a mechanism here now for propagating an electrical current along this axon. And so we're going to watch a little video here about how this works. But this is just a cartoon diagram of a nerve cell. Nerve cells come in different sizes, different shapes, but they share certain features. There is a cell body. This is where you find the nucleus, most of the organelles. There are processes on that cell body called dendrites. They are going to receive electrical signals from the environment. Then there is a long process, variable length, that can go anywhere from a millimeter, a thousandth of a meter, to more than a meter in length, depending on which axon we're talking about here. And that is going to conduct electrical current in the form of ions. It's going to propagate an action potential. And then that terminates in a set of processes that form the actual nerve terminals here, where the signal is then going to be transferred to another excitable cell, either a muscle cell or to the dendrites of another nerve cell. But let's go ahead and watch this. First, I need to make sure that the sound is on here. And we want to turn that.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. To transmit an action potential over such a distance without weakening requires that the signal is continuously re-amplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state, in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize, away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium ions to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. The action potential is propagated along the length of the axon in only one direction. By examining the membrane potential and state of the sodium channels along the length of the axon, we can see why this is so. As a depolarizing stimulus reaches our section of the membrane, sodium channels open and current flows into the axon. This in turn depolarizes adjacent sections of the membrane, causing adjacent sodium channels to open, and the action potential is thus propagated along the axon. Sodium channel inactivation prevents the depolarization from spreading backward along the axon.
Okay, so that makes sense to everybody. That is, we focused up to now on just what's happening with a single sodium channel, but that sodium channel is right next to some more. And so once the first one gets set off, it depolarizes the membrane around it, which causes the next bunch to open, and then they become refractory to reopening, but by that time they've triggered the next bunch. And so what you have is this wave of sodium moving down the length of the axon, and it can only move in one direction because the sodium channels behind are refractory. They can't reopen again until something depolarizes the membrane around them again. And so electrical signals, electrical current flow in one direction along this nerve cell. Always flows from the cell body toward the axon terminus. And of course, you've got different nerves. Some of them are outgoing, some of them are incoming. Some of them are carrying signals from the central nervous system to the periphery of the organism. Some are carrying them from sensory receptors in the periphery toward the central nervous system. And they're just oriented in opposite directions. There's a player in this whole thing that this particular animation leaves out. And I want you to know about it, but you aren't responsible for it. It just complicates the whole thing. There is, in addition to the voltage-gated sodium channels, a set of voltage-gated potassium channels. And those channels open up right after the sodium channels have become refractory to opening. That is, you open it up, the sodium rushes in, and then those guys get blocked and no more sodium can come through. And then right at that point, you open up these potassium channels. Well, remember, the potassium was concentrated inside the cell, and not only that, but because we've just depolarized the membrane, there is a big electrochemical gradient pushing that potassium out. It wants to go back outside because now it's negative outside relative to inside. So that potassium flow helps drive that repolarization of the membrane that we saw. The sodium pump is also involved, but so are these potassium channels. I just want you to know they exist because you'll hear about them in other classes, and you say, Dr. Linn never told us anything about voltage-gated potassium channels. He only talked about sodium channels. They do exist. They're just not in this particular diagram. But they help to accomplish the same thing, a wave of positive charge moving down the axon of a neuron toward the termini of the cell. Come on, little friend. Oh, now we're stuck. There we go. What happens when our nerve impulse, when our electrical sodium current gets to the terminus of the neuron? Well, you reach a synapse. The synapse is going to be located at one of the branches of the neuron in the terminal region. And in a synapse, you have got vesicles filled with chemical nerve transmitter. Those nerve transmitters are going to be ligands for something. They're going to be ligands for ion channels. These things are clustered just under the plasma membrane here at the terminus of the axon. In the membrane of the terminus, there's going to be a new set of channels. They're calcium channels, and they're voltage gated. And in the absence of an action potential, they're closed. But now the action potential comes flowing down the axon. It gets to the terminus in the synaptic region. The membrane becomes depolarized. That causes this voltage-gated calcium channel to open up. There is excess calcium out here in the extracellular fluid relative to its concentration inside. So that stuff's going to go through our calcium channel. We've got some positive charge moving from outside the membrane to inside the membrane. We already saw what happens there with our sodium channel. We're going to depolarize the membrane. When we depolarize the membrane here, that is going to trigger the fusion of these synaptic vesicles with the end plate of the synapse. When they fuse, what was inside gets released to outside. So now this neurotransmitter is out here in the environment just outside the plasma membrane. And there it's going to encounter ligand-gated channels on the membrane of something else, muscle cell or another neuron. And it's going to bind to its specific site. And that's going to trigger the opening of this ligand-gated channel. And it's quite often a ligand-gated sodium channel. Well, sodium concentration, as always, is kept higher outside the cell than inside. So you open this ligand-gated channel, sodium flows in. What's that do to the membrane potential in the next cell? It's going to make it more polar or less polar? Who says more? Who says less? The less wins, okay? So the sodium, high concentration out here, is going to rush into the cell. And that is going to reverse the polarity just as we saw before. Now guess what? Around our ligand-gated channel, there are going to be voltage-gated sodium channels. And they're going to be triggered to open. So now we see how you got that action potential started in our neuron in the first place. That is, ligands binding to their particular receptors, which were, in fact, ion channels, cause those channels to open, depolarize the membrane around them, triggering the opening of voltage-gated sodium channels. boom, start a new action potential. And so this is how you get a signal from here to here to here to here to here. Sequential opening and closing of voltage-gated channels and ligand-gated channels. And one of the most important ligand-gated channels is represented here. This is the acetylcholine receptor. Anybody know what acetylcholine is? Yes. It is a neurotransmitter, correct. And it's a neurotransmitter especially that is important in triggering what? Anybody know? Contract of what? Muscle contraction, that's right. And so the plasma membranes of muscle cells are going to have acetylcholine receptor in them. Acetylcholine receptor is a multi-subunit protein here. It is integral in the membrane, and it has ligand binding sites for its neurotransmitter acetylcholine. The other thing it is it's a sodium channel. It's a ligand-gated sodium channel. And so when an action potential from the brain comes down a motor neuron toward the muscle end plate, you trigger the fusion of acetylcholine vesicles with the nerve terminus. Acetylcholine gets released into the space of the synapse between the muscle and the motor end plate of the neuron, and that acetylcholine binds to its sites here on the receptor. That is going to trigger entry of sodium in the muscle cell, meaning that that membrane just got depolarized. And when it gets depolarized, it causes the opening of voltage-gated channels around it. And that's going to depolarize that muscle membrane completely, and that's going to lead to the opening of calcium channels in the sarcoplasmic reticulum, like we saw with the ryanodine receptor 2, which is found in heart muscle, not skeletal muscle. There's a ryanodine receptor 1 that you find in skeletal muscle. But then that's going to cause the opening of that channel in the calcium storehouse. Calcium is going to rush out, and as you'll see later on in the course, that's going to trigger muscle contraction. And so this is a directionally coordinated system of conducting electrical current in the form not of free electrons bouncing from atom to atom in a metal wire, but rather positively charged ions moving down the length of an axon in a nerve cell until they reach the terminus. So here's what you should know about ion channels and electrical gradients. What are the structural characteristics of ion channels that they all share? They all have a single gate. They all have that selectivity filter that is going to determine what charge and what width you can be and still get through. They are going to be integral membrane proteins, usually composed of multiple subunits. What is membrane electrical potential? It's the difference between the charge on one side of the membrane and the other, and we define it as the distance in which a positive test charge would move. If we say that a positive test charge would move from outside the cell to inside, then the cell has a negative electrical potential. It's usually measured in a range of minus 20 to minus 200 millivolts. The animation quoted a figure of minus 80. Again, that's within that range, but don't get hung up on that. That's not what they all are. It depends on exactly what membrane we're talking about. But it is, again, the direction in which a positive test charge is going to want to move, and for cells in an unstimulated state, it's going to be negative. Resting membrane potential is what the membrane potential is when none of the channels are open, okay, when no electrical current is flowing in. What produces the cell's resting potential? Sodium pump and those potassium leak channels. What's depolarization? It's when you remove that normal polarity, and in fact you usually reverse it. You go from being positive outside to negative inside to being negative outside and positive inside. The distinctions between the ligand-gated, voltage-gated, and mechanically gated channels, what causes them to open? What's an action potential? It's an all or none depolarization. It's either completely open or it's completely closed. And how do you start one? Well, you got to open up a few channels and cause a local depolarization of the membrane, which is then going to cause the surrounding voltage-gated sodium channels to open. And it's going to spread in a directional fashion because once one of those channels opens, it quickly becomes plugged by that tethered domain that puts it in its refractory period where it can't reopen again for a particular period of time. How do you spread action potentials across synapses? By getting vesicles filled with neurotransmitter to fuse with the motor end plate, or the if you've got a synapse between one neuron and an interneuron, between the dendrites of one and then the processes at the terminus of the other. How does the acetylcholine receptor work? It's a ligand-gated sodium channel. So you want to know all of those things. Any questions about ion channels, what they do and how nerve impulses work? Yeah? Okay, so those, I talked about the leak channels before, which are not gated at all. They just open and close spontaneously. But I also mentioned those voltage-gated potassium channels that contribute to repolarization. So they're part of it, and the sodium pump is the other part of it. Okay? The potassium channels are actually important because opening and closing of channels happens very, very fast. Whereas the time necessary for the sodium pump to move all that sodium back to the outside, that takes longer. So it's more of a long-term factor in the repolarization, whereas the voltage-gated potassium channels are an immediate thing. So that's why you see that potential go up, spike, and then immediately start to come down. When you get up to that spike point, that's when you open up the voltage-gated potassium channels. Now the potassium begins to flow out, and that causes polarity to return back toward where it was. And then the sodium pump just completes the process. Is that okay? Anybody else? Yes? What is the reason for it, the stoichiometry? That's just the way that the pump is designed. It's called an electrogenic pump because it does, in fact, create a membrane potential, because it doesn't move an equal number of positive charges in and out. There are other pumps that are non-electrogenic, one for one, positive this way, positive that way, negative this way, negative out that way. But the sodium pump has binding sites on the interior side of the cell for 3 sodium ions at a time. And it has binding sites for 2 potassium ions on the other side. So it binds its 3 sodiums, and then, remember, it's a transporter here, so it's got 2 gates. You close that inner gate, and that opens the outer gate, allows 2 potassiums to bind, then you hydrolyze your ATP, boom, you actually move the sodium ions in one direction against their concentration gradient, and the potassiums in the other direction against their concentration gradient. So why does it work that way? Because that's the way the protein is actually evolved to function. Yes? Remember the potassium leak channels. Those are always opening and closing, opening and closing. And there are no comparable sodium leak channels. Well, I shouldn't say that. There actually are such things as sodium leak channels. There are. But there are very definitely potassium leak channels that contribute to resting membrane potential. And again, they're just spontaneously opening and closing. And because the sodium pump is concentrating potassium inside the cytosol, then it's going to want to tend to flow out if you give it a way to do that. And that is contributing to that resting membrane potential. So both the sodium pump and the potassium leak channels contribute to that. Any more? Okay, I hope I didn't miss anybody. Well, in the time we have left, we're going to change gears completely. And we're going to start, whoops, wrong way, talking about cell energetics. And by energetics here, we mean metabolism. And specifically, how cells extract energy from fuel molecules and transform it into a type that they can readily access to do work. Chemical fuels are the energy source for most of the organisms that we tend to think about, certainly for ourselves. For any other type of animal, for many types of bacteria, chemical fuels are going to be the source of energy. Now, for others, it's going to be light energy. Plants, the algae, photosynthetic bacteria, all of these guys can directly capture light energy and transform that into chemical energy. But a lot of the organisms here on Earth have to get chemical bond energy to begin with. They cannot use light. Now, you can release energy stored in chemical bonds in different ways. You can take a sugar, it could be glucose, sucrose, fructose, any sugar, and you can actually burn it. In chemistry class, you can do some flame photometry or something like that, and you can actually light this stuff on fire, and you can measure how much energy is released when you do that. And if you burn a carbohydrate in the presence of oxygen, you have very large activation energy barrier you have to overcome. That's provided by the flame, a lot of thermal energy there. And that will result in a free energy change that gets you to carbon dioxide plus water. Those are the products of complete combustion of a sugar molecule. So the sugar and the oxygen together, they have a certain free energy level. The carbon dioxide and the water, they have their own free energy level. And the change between the one and the other is a fixed quantity at a particular set of environmental conditions, particular temperature, pressure, so on and so forth. That is, that's inherent. Now, you can quote unquote burn sugars in a living organism too. You can release the chemical bond energy. And you can convert sugar all the way to carbon dioxide and water. And you know what? The free energy change is exactly the same. The total amount of energy that you can get out of that sugar when you oxidize it all the way to CO2 and water is exactly the same as you would get if you burn that sucker. So why don't our cells do that? Why not just somehow have a little electric spark in each of our cells that sets that sugar on fire and just releases that energy of chemical bonds in one glorious burst? Why not do that? It would be like what? It would be very hard to control, and how would most of the energy be released? Heat. Yeah, you all are getting used to that one now. And can cells use heat to do work? No, so it would just be radiated out into the environment and nothing would get done. And so cells don't want to lose all that energy, and they want to be able to control things, which is absolutely right. So rather than releasing the energy from the chemical bonds of fuel molecules all in one big step like you do in combustion, you do it in a whole bunch of small steps. And those small steps are catalyzed by enzymes. And of course, enzymes lower activation energies. They lower the amount of input thermal energy you need to get a reaction to start, meaning that we don't need something like a spark to actually trigger energy release. And so cells break down fuels in multiple steps, and along the way, since they're not releasing all of that energy as heat in one big boom, they're able to capture some of it, and they store it in other chemical bonds. Now, they don't store all of it. First law of thermodynamics says, no, you can't do that. First and second, I should say. You're always going to lose some to entropy. But you can capture quite a bit of it this way, which you couldn't do that way. Notice something, though, about the free energy change, going from sugar to CO2. It's exactly the same in both cases. That is, you can get the same amount of energy total out of burning a sugar molecule as you can from metabolizing it in a cell. It's just the way that you go about releasing that energy is different. And that allows you to capture more of it, and it also allows you to control the process so that you're releasing exactly as much energy as you need when you need it. The process of completely oxidized, oh, I'm sorry, yes. I couldn't hear the last thing you said.
Is it still considered a combustion reaction?
It's not combustion, really. I mean, we talk about it sometimes that way. You know, we say our bodies are burning fuel. We're not actually carrying out a combustion reaction, but the combustion reaction releases exactly as much free energy as a non-combustion enzymatically catalyzed pathway does. You can't do anything about that free energy change. It is what it is. Respiration is the oxidation of fuel molecules all the way to CO2 and water. And respiration has several stages to it. Now, in this particular schema, I'm going to show you 3 stages are cited. If you consult other sources, they'll talk about 4 stages, and I'll show you where the difference is. We're all talking about the same thing. The first stage of respiration involves taking complex molecules, proteins, nucleic acids, fats primarily, and polysaccharides, and breaking them down to their constituent subunits. The reason you do that is that proteins can't get across cell membranes very well. Fats can't get across cell membranes very well. Polysaccharides can't get across cell membranes very well. So you got to break these complex macromolecules down into smaller bits. Those can be moved across the membrane by transporters. So stage 1, the breakdown of foods to simple subunits, occurs outside cells for the most part, with the exception that some fuel molecules can be taken into cells through the process of endocytosis. Now, we haven't talked about that yet. We're going to talk about it later. Does anybody know, just jumping ahead, what endocytosis is? Yes? Okay, that's part of the pathway. But what is endocytosis itself? Somebody I haven't called on yet. Somebody back in the back somewhere. Anybody give us a quick explanation of what endocytosis is? Yeah. Okay, is that how they bring all things inside? Okay, what kinds of things? Okay, pinching off of the membrane. Very good. That is endocytosis. And what kinds of things do you bring in that way? We already looked at transporters. You know, and transporters can bring in things like sugars, amino acids. That is, all the breakdown products of these of these macromolecules. But what about endocytosis? What kinds of things can that bring in?
Charged molecules?
You could, but that's not what I'm thinking about. I'm thinking in terms of size. Transporters can bring little things in. What about endocytosis? You're actually wrapping the membrane around something. Yes. You could bring in other cells like a bacterial cell. Or you could bring in a glob of fat. Or you could bring in a chunk of polysaccharide. That is, endocytosis is a way to bring in big stuff. Then you get to what somebody else said. You can break these things down in the cell. And you do that by getting the vesicle that pinched off from the membrane to fuse with a lysosome, which is full of hydrolytic enzymes, which break these things down into small subunits that you then transfer out of the lysosome into the cytosol. So either you break it down outside the cell and transporters bring it across the plasma membrane, or you bring it in through endocytosis, you break it down by fusing with a lysosome, and then you still get those small bits out into the cytosol. So that's stage 1 of respiration, just getting the stuff into the cell. Stage 2 is going to be taking those simple subunits and breaking them down to something called acetyl-CoA. Acetyl-CoA is a 2-carbon molecule. So however big this thing was, you're going to reduce it down to these little 2-carbon bits. Along the way, you're going to produce some ATP. ATP is the universal energy currency of cells. This is kind of our goal. This is the activated carrier that lots and lots of biochemical reactions in cells are going to be able to use. You're also going to produce another activated carrier, NADH, nicotinamide adenine dinucleotide hydride. And that is going to carry high energy electrons that can be used later on to do work. So both of these things are going to occur in stage 2. Stage 2 involves two specific pathways. One is glycolysis, which we'll look at in detail, and that's where you make some ATP and some NADH. Glycolysis forms a 3-carbon molecule called pyruvate, and in a eukaryotic cell, pyruvate is then going to be transferred into the interior of a mitochondrion. And in the mitochondrion, that 3-carbon molecule is going to be broken down to a 2-carbon molecule. The 3rd carbon is going to be released as carbon dioxide, completely oxidized carbon. And in the process of making acetyl-CoA from pyruvate, you get another one of these activated NADH molecules. What if you're a bacterial cell and you got no mitochondria? Can this happen? I heard somebody say, yeah, she was right, wherever she was. Yeah, so what happens there? I mean, you got no mitochondrion to go into. So where does the conversion of acetyl-CoA, or of pyruvate to acetyl-CoA happen if you're a bacterial cell? Yeah. Not in the cell membrane. In the cytosol, yeah. The cytosol of a bacterial cell or a prokaryotic cell is analogous to the inner environment, the matrix of a mitochondrion. Same reactions that would happen in the mitochondrial interior happen in the cytosol of a bacterial cell. There's another type of pathway we'll talk very briefly about called fermentation. Fermentation is not respiration. Respiration is completely oxidizing your fuels, getting all the energy out of them that you can by converting them to CO2 and water. Fermentation is partial oxidation. Fermentation involves stages 1 and 2 of respiration, just breaking down the big stuff, bringing it into the cell, and then carrying out glycolysis. And then it stops here. Almost. And we'll talk about what the almost means. Stage 3, and in some schemes, stages 3 and 4 of respiration occur in the mitochondrion if you're a eukaryotic cell, in the cytosol if you're a prokaryotic cell. And stage 3 is going to involve the oxidation of that acetyl-CoA that you made all the way to water and CO2. For those sources that separate things into a stage 3 and a 4, whereas this particular one combines them, what happens in stage 4 is something called oxidative phosphorylation. And in oxidative phosphorylation, you take activated carriers, specifically NADH and another high-energy electron carrier called FADH2, that you made in stage 3 and in stage 2, and you use those high-energy electrons to do work in something called an electron transport chain. And the electron transport chain allows you to make lots and lots and lots of ATP from the chemical bond energy that those electrons carry. And so the net result of stages 1, 2, and 3, and in some cases 4, which in this scheme here, they're lumping 3 and 4 together, is to take food molecules and molecular oxygen, because we're talking about aerobic organisms here, and make from that ATP, NADH, FADH2, which they conveniently left out in their figure, but I put it back, and then CO2 and water. So complete oxidation of fuel molecules to CO2 and water, and capturing as much energy as you can capture in the form of these activated carriers, ATP, NADH, and FADH2. Stage 1 happens outside the cell or in lysosomes. Stage 2 happens in the cytosol, whether you are a eukaryotic or a prokaryotic cell. Stage 3 and or stages 3 and 4 happen in the mitochondria if you're eukaryotic, and still in the cytosol if you're prokaryotic. So you want to know what those stages are and where they occur. So here's the summary. Cells release energy from fuel molecules in small sequential steps, not all at once. We said that's because they can capture more energy that way, and they can control the process. There are 3 or sometimes 4 stages in cell respiration. Know what they are and where each one occurs and what happens in it. In cellular respiration, you completely oxidize organic fuels. What are the end products? Well, you should know, CO2 and water. CO2 is the most oxidized form of carbon. You can't get any more bond energy out of the molecule at that point. And then finally, this other phenomenon, this partial oxidation strategy, fermentation, is different. It doesn't have a stage 3 or a stage 3 and 4. It only has a stage 1 and 2. And so you capture some energy, but not all the energy that you could potentially get. Why would a cell want to carry out fermentation? Can anybody speculate on that? You know, why, if you're not getting all the energy that you could get out of it, why even do it? Because of what? It's faster, yes, because you don't do as many steps, but that's not actually the reason. Yes? Oxygen. Oxygen. In our respiration here, which is aerobic respiration, oxygen is absolutely necessary for this process to take place. If you got no oxygen, you can't do it. Can't do it. Now, there is such a thing as anaerobic respiration, and we'll talk about that. But you can't carry out this scheme without oxygen. So that's one reason to do fermentation. Anybody think of another one? Yes? Oh, I think you said exactly what I was thinking. Can I come up so I can hear you better? Well, okay, they don't have mitochondria, you know, if you're a bacterial cell, but it gets more to something simpler, more basic. You don't do it because you can't. You don't have what you need. You don't have the enzymes necessary to carry out all of these steps. You only have some of them. And so you settle. You settle. You're a simpler organism. You have the ability to carry out the breakdown of macromolecules to smaller subunits. You can carry out glycolysis. You can get a little bit of ATP and NADH that way, but you can't go any further because you don't have the enzymes to do it. Or you have the ability, but you're in a place where there's no oxygen. So there are reasons that organisms ferment instead of carrying out respiration, either because the conditions limit them to doing that or their own genetic makeup limits them to doing that. Don't everybody jump up yet. According to my clock, we've still got 4 whole minutes to go. Whoops. Let's do an overview of stage 2, glycolysis. Now, this isn't biochemistry class, so I'm not going to expect you to memorize the intermediates. I'm not going to expect you to memorize the enzymes. I'm not going to expect you to memorize the structures. What I want you to know is what happens. What happens? What does the pathway accomplish? Where do you begin? What do you end up with? And what are the big steps between the start and the finish? And that's what you're going to be responsible for. You take 342, oh yeah, you're going to have to memorize a lot of this stuff, but you'll be ready for it then. What I want you to do is get the big picture now. And for those of you that already went through biochemistry, I apologize to you're getting the Cliff Notes version now. Glycolysis uses glucose as its starting material. Glucose is the preferred fuel, if you will, of living cells. It's the preferred fuel because they can get the most bang for their buck from that fuel. They can use lots of other fuels, but they have to spend a little more energy to do it, so their overall yield in the end isn't as high as it would be if they used glucose. In glycolysis, you take a 6-carbon sugar molecule and you split it into 2 3-carbon molecules. They're called pyruvate, pyruvic acid in its protonated form. Along the way, you actually have to spend some ATP. So, oh, hold the phone here. I thought we were talking about energy extraction here. What in the heck are you doing burning ATP? Well, you got to use a little to get some more. ATP is going to be invested in glycolysis to raise the free energy of the intermediates to a level in which you're able to carry out reactions that otherwise wouldn't go energetically. After creating 2 3-carbon molecules from our 6-carbon molecule here, we're going to get from each one an NADH, that is an activated carrier of electrons, and we're going to get 2 ATPs from each one. So putting together our 2 3-carbon molecules, we got 1, 2, 3, 4 ATPs and 2 NADHs. We have to subtract out the 2 ATPs we invested. What's our balance sheet? 2, 2, 2. Yes, easy to remember. You get back a net of 2 ATPs and 2 NADHs from your glucose molecule. All of these reactions occur in the cytosol. They involve soluble enzymes. And oxygen isn't used anywhere in this process. So this explains why our organisms that are stuck in an environment with no oxygen can use fermentation. Fermentation is glycolysis plus a couple additional reactions, and we'll get to what those reactions are later. But this is a way to get some ATP without having to use oxygen. And we will continue on the path of glycolysis next time. Okay, so you're thinking